Good morning. Welcome to the Rotary Club of Hudson. My name is Marilyn Orr, President of the club, and we are thrilled to have you join us today. This meeting broadcast is available on HCTV, as well as our Rotary of Hudson YouTube channel and Facebook. Our club meets weekly using this digital platform, Zoom. When we meet in person, it's at Laurel Lake Retirement Community for breakfast. We hope to get back to that soon. We meet most Wednesday mornings from 7:15 to 8:30 a.m. So come out to join us for a meeting. We would love to have you as a guest. To learn more about Rotary, our club, and the impact we are having on our local community and the world, please visit our website, rotaryhudson.org. So enjoy this meeting today and help us share the message that Rotary opens doors of opportunity. All right, and we are ready to go. Good morning, Rotarians, and welcome to our meeting. Today is January 20th, Inauguration Day, a day of new president coming in and a big day in our nation's capital. We're going to start today with our invocation by Dan Williams. Dan, please unmute yourself and let's get started.
I think I'm unmuted. You can hear me?
Yes, I can.
We're good to go. Let us pray. Lord, thank you for allowing us to gather as Rotarians on this significant day in our country's history. We ask that you would provide protection for our new president and vice president, that you would also provide protection for our nation's capital and all the people in the DC area today. We ask that you give President Biden and his administration wisdom to know how to control the virus that is gripping our nation. Please help the companies that are producing the vaccine to be able to meet the demand levels necessary to control this virus. And finally, we thank you for allowing us to live in a great country like the US and ask that you provide the guidance to help us return to some sense of stability. For we ask all these things in your holy name. Amen.
And for the four-way test and our Pledge of Allegiance, Terry Bortnick, would you please unmute yourself?
Oops, can you hear me now?
Yes, ma'am.
Okay. I pledge allegiance to the flag
of the United States of America
and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all. And the four-way test: is it the truth? Is it fair to all concerned? Will it build goodwill and better friendships? Will it be beneficial to all concerned?
Thank you, Terry. How wonderful. We're going to start this morning and jump this meeting off with Jamie Cosman from the Hudson High School Service Learning class. Jamie, let's introduce the students first, and then we'll view the video that's been prepared.
Sounds good. So good morning, Rotarians. So good to see everybody. I wish we were in person, and I look forward to that day when we do return to that. It's been way too long. But this morning I have the pleasure of introducing both Alyssa and Kate, who what I think is so fascinating is I think they give us such a great experience or a great opportunity to look at two very different service experiences, even though they're both volunteering technically in the same school. They're both over at EWIS right now. We also wanted to let you know that applications are currently out for next year's class. They are coming in as we speak, and they're due Tuesday by 3 o'clock. So if you know any juniors in the high school who would be good for service learning, please push them that way. And that extra push can be so meaningful and powerful. And we also wanted to let you know that this is actually their last week at their current first semester site. Technically, we started the second semester the other day, but with us having kind of an awkward schedule this week, we decided to start next week with the new site when it's going to be a more regular schedule. So both of these, both Alyssa and Kate, will be at a new site starting next week. This year has been, as you know, a year where I think the key word is flexible. And I'm so grateful to our students because they have been beyond flexible, beyond willing. We had 15 students who were serving remotely during the first semester. We were able to get that number down to 5. Kate was one of those students who was serving remote, or is currently serving remotely. And yet we never heard her complain, and we never heard them complain, which I think is such a tribute to them, because service is so often, and when students are joining service learning, they're so often looking for that hands-on in-person experience. So their willingness and their flexibility to help make this work is something to really be commended. And without further ado, I'll turn this over. I forget if you said you wanted to go to the video or if you wanted to go to Kate and Alyssa first.
Let's hear from the girls first.
Yeah.
Okay.
I'll go first. Hi, I'm Alyssa. I'm also in the service learning class. For my site, for first semester, I was able to be at EWIS, so Eastwoods, with the 4th grade, helping a teacher with a 4th grade gifted English class. And in this English class, I basically was able to, it's a new year for everybody. No one's used to the schedules we're currently operating on, or obviously we all got introduced to Zoom this year. So I feel with the teacher, with my teacher, I've been able to help a lot in managing that with the students and the coursework. And I basically go around and help the teacher with whatever she needs. We create a lot of lesson plans for the class to keep them interactive during this time. I've been able to pull classwork that we've done in service learning at the high school, like finding your spirit animals and personality tests, and suggest that to do it in our classes at the 4th grade level. And so we've been able to build a social emotional project around that. And basically I just help around, walk around constantly, check if anyone needs help, because I know a lot of students these days are confused on what even is happening or what days to go to school. So I think it's been really good for the kids to have 2 people in the classroom. And my teacher's also managing all online kids as well. So she basically has to teach 2 different curriculums within one classroom, which most teachers don't. So we can switch roles. I can take charge of the Zoom people or the people in person. So I think that's just been able to help the classroom progress as efficiently as possible with this, obviously Corona and everything. So, I mean, for me, this experience has been so positive. I mean, it's been positive just simply because I've been able to go out and serve, and that's exactly why I joined service learning. And I think it's made me a better person in general, just because I've been able to get that sense of also increased responsibility. I mean, I have to dress like a teacher has to dress. I have to speak to the kids in a manner like a teacher would have to. And just managing, my supervisor that I'm with, the teacher, is currently quarantining right now. So I'm basically in charge of the classroom a little bit, because she's online on Zoom, but that only goes so far. It's hard with communication and everything. So now I'm definitely feeling that increased sense of responsibility, because I'm basically the only teacher in the class now, which has been interesting, but I actually like that I've been given that opportunity too. And just, I've had to keep in mind all those things, patience. And I also now better understand why the teachers are probably what they're more experiencing on a daily basis with these Zoom calls, how frustrating they really can be. So just, I think now, like, I don't really like, I understand a little bit what teachers are talking about now when they say this time has been difficult, because I've been able to see it firsthand with my supervisor. And Zoom really can be hectic sometimes, but I think like throughout this time, just like Mr. Cosman said, just to be flexible and realize like we are fortunate at this time, and um like we're still able to go to school. Basically now we're going into all in soon. So just to be, there's no point in a way dwelling on the fact of, oh, I'm hybrid, I can't go to school half the week. It's more just I can still go on Zoom calls, which is great. And I can wake up later too, which is a positive to look at when I can go on Zoom calls. So yeah, just it's been important, I feel like, this year to be flexible and realize also like teachers are doing what they can as well. And I think that's really, the site has really helped me see that. So yeah.
And Alyssa, what do you plan to do after graduation?
So I used to live in Europe my first until I was 8. I moved to America. So now I'm planning to go back to Europe for college. And I mean, the whole application process is a little bit later for European school. So I'm still waiting to find out and doing college interviews still, but I'm planning to go to the Netherlands for a liberal arts and sciences college, and most likely go into political science or psychology. So yeah, I'm really excited for that. It's going to be a big change, but it's also going to be a learning experience as well. So
Hi, I'm Kate. I'm also in service learning, and I served at US as well, but it was all remote. So I served with my supervisor, and she was like an aide who we kind of helped third, fourth, and fifth graders outside of their normal classes. So some kids who needed just a little more help, or also some autistic kids as well. And these are kids that are fully online for the whole year. So it was definitely harder to try to make like a connection with them over Zoom. But over these past few months, I feel like I've made just as great of a connection with all of these kids that I worked with, even the fact that I wasn't actually there with them. But yeah, so I kind of did similar things that Alyssa did. We made like lesson plans. I did a lot of one-on-one things because that's the easiest thing we can do with Zoom for whatever the kids need help with. Sometimes we were one-on-one with the kids, and other times we were in smaller groups. But kind of just, we helped like get them to where they need to be, because I learned a lot on like in Zoom that these kids, some of these kids have a harder background at home. And it was actually really eye-opening, because you wouldn't see that if I was in school with them. And just being like, even in Hudson, we kind of find that we're not always being able to see like where these kids come from around in like their home. And I just thought that for me, it was really eye-opening, because I wouldn't have been able to see that if I was in the classroom with them. But yes, service learning has been a great opportunity for me. And I've done Hudson Community First and things like that throughout the community. But this is kind of a way for us to get outside of Hudson. For my next semester, I'm outside of Hudson, so I think it'll be really exciting for us to be able to do something outside of our community as well.
And what are your plans after high school?
I'm not sure where I'm going to attend college, but it's probably between Ohio State and Miami University. And I'm kind of still deciding my major, but I want to become a sports broadcaster.
Okay, James, let's see if we can tee up the video. And thank you. That was wonderful to hear about everything going on at the school. It'll take just a moment as we get this going.
Does everyone see a big pink screen?
Yes, yes, we're good to
Okay.
go. We're not getting any audio, James.
One second, there's something I have to do to get the audio going.
So when you share this screen, there's probably two little boxes in the bottom left corner of the screen, the pop-up window. If you click both of those, it should maximize the audio for the video.
Okay, let's see. Here we go. Share sound. Very good. Okay.
Just gonna stand there.
Can we hear that now?
It's definitely more difficult for us to be able to serve outside of Hudson and help others beyond our community. But I think a lot of us have learned this year that the smallest things we do can help others in such a big way, more than I think we'll ever know. I think that's kind of what service learning is all about, just being able to find service and helping others and putting it into our everyday lives and making strong and lasting relationships along the way.
My favorite thing about service learning is going out of Hudson and doing something I've never done before and meeting new people in my class that I haven't had classes with ever, which is really fun to have people senior year that I've never really talked to.
What I like most about service learning is the teachers. They're the best. They motivate you to be the best student you can be and community servant you can be. But then also the aspect of serving in our community. I know that I'm making a lasting impact on our community and others around me.
My favorite part about service learning is that everyone is so open and accepting every day when you walk into class, and all your teachers want to see you succeed as you gear up for college and the rest of your life. So join.
My favorite part about service learning is getting to meet all the new people that I normally wouldn't have a chance to talk to in other classes, and the teachers, because they really just set you up for success and they really care and they help prepare a lot for college.
My favorite part about service learning is getting to go to site 2 days a week and spend time with all the little kids there.
My favorite part of service learning has to be talking to new people that you would never talk to in the past and creating new relationships with them, and also how supportive all of the teachers are.
My favorite part about service learning is going to my site.
The best part about service learning is probably the experiences and people you get to meet outside of Hudson, like at your site and stuff that you would never be able to meet if it wasn't for this class.
What I like most about service learning is the flexibility of the teachers and the schedule. You know, things change day to day, and the teachers have done a really awesome job meeting the needs of the students and making the class feel like a college class.
Hi, my name is Meaghan Lidl, and I'm from the service learning class of 2020. My favorite part of my service learning experience was easily the teachers. Mr. Bach, Mr. Cosman, Ms. Omni, and Ms. Bonville did a great job of making every student feel so loved and so supported through no matter what they do, even if it's outside the classroom. My favorite days at site were when they'd come and visit and I'd get to show them what I'd been doing for this past couple weeks. And I felt like I could tell them anything. Even after I've graduated and I've moved on from the service learning program, they're always such a steady rock that I know I can go back to if I ever need to talk to.
it's meant to be, it'll be, it'll be. Baby, just let it be. If it's meant to be, it'll be, it'll be. Baby, just let it be. So won't you ride with me, see where this thing goes. If it's meant to be, it'll be, it'll be. Baby, if it's meant to be. No darling, no sleeping, live it up like it's the weekend. When the DJ play the right song, gonna drink, gonna party all night long. No darling, no sleeping, live it up like it's the weekend. When the DJ play the right song, gonna drink, gonna party all night long. I can feel your love pulling me up from the underground. I don't need my drugs. We could be more than just part-time lovers. I can feel your touch picking me up from the underground. I don't need my drugs. We could be more than just part-time lovers.
There we go. Obviously, someone has got serious editing skills. Wow, that's incredible. Thank you so much, students. And thank you again, Jamie, for bringing this program to us this morning.
Hey, Marilyn, can I ask Kate a question? Kate,
Sure.
are you going to play basketball in the Netherlands?
That's Alyssa.
Oh, that was Alyssa.
Play basketball though. Yeah.
Oh, okay.
All right. Thank you, everybody, so much for having us. It was great to see you and to catch up with you. And I look forward to seeing everybody last month. Did you have any questions for us before we leave?
Jamie, I would just suggest that we present this video at the beginning of one of the board meetings. It's really fantastic.
Yeah, you're welcome to. You've got access to it now through YouTube. So if you need the actual file of it, just let one of us know, and we can definitely get you the actual file too.
There we go. Really nice. Well, we're going to jump into our program this morning.
Bye, everybody.
Okay, bye-bye. Have a great school day.
Thank you.
So good
Bye,
morning.
thank you. Have a good rest of your day.
Okay, here we go. Meaghan Russell is a Hudson native and WRA graduate. She obtained her bachelor's degree in biology, MPH in epidemiology and biostatistics, and PhD in molecular and cellular biology and biochemistry from Boston University. She followed that with postdoctoral work at Harvard Medical School. Her doctoral and postdoctoral research was focused on the genetics of congenital anomalies, specifically diaphragm and lung defects. Following her postdoc, Meaghan transitioned to the cancer diagnostics industry, where she has spent the last 8 years leading the development of variant interpretation and clinical reporting platforms to provide cancer patients with personalized treatment options. Meaghan currently works at Invitae as the clinical genomics lead of somatic development. Meaghan lives north of Boston with her husband and 2 boys, James 7 and Cole 5. She enjoys traveling, especially back to Hudson to see her mother, past president, Allison Feaster. She enjoys hiking and spending time with her family. So Meaghan, would you please unmute yourself and let's get going.
All right. Well, thank you so much for having me. It's great to be in Hudson, even if it's by Zoom, but wonderful. So I'll just get started. I'll share some slides right now. Let's see here. All right. I might end up having to. All right, present. All right. So I'm gonna give a couple slides and feel free to stop me along the way. I won't be able to see you, so just interrupt anytime, it's no problem. And I'll just talk about the work I do for trying to understand how we can use our genomics to try to determine what therapies patients can have when they have cancer. And how we identify personalized medicine is really, it involves an individual's clinical information or genetic profile, meaning that we do sequencing on the patient's DNA and we identify all the genes as well as the variants that are associated with particular dysfunction. And then we're able to target those particular variants with drugs. And so that's really the goal of what we try to do with specifically with personalized medicine, but we can also learn about diagnosis, prognosis, or other aspects as well. And I'll give you a brief Cancer Biology 101. And basically, we talk about cancer and how it's caused by the hallmarks of cancer. And it's the cancer field, it's very rapidly evolving. So that every couple of years, we add in new ways where we learn about how cancer is caused or in combination with other different systems. And when we think about cancer, we think about the biological capabilities that are acquired in cells that then can develop tumors. And we really think about how cancer cells can keep on proliferating or growing, and they can signal to other cells to make more and more proteins that then directly are associated with cancer. They can evade growth suppressors. So we'll have genes where there could be a mutation where typically it holds things in check. And then if you have a mutation, then all of a sudden things can keep growing. Cancer cells are able to resist cell death. They can replicate very quickly. They induce angiogenesis, meaning that they're able to vascularize tissues, tumor tissues, because they need the oxygen, they need the nutrients to grow and to be sustainable. And they can also they activate invasion and metastases. And more recently, the field has been exploding in terms of immunotherapy, where we realized that cancer can hijack or take over the immune system. And so now there are new targeted therapies to handle that, as well as genomic instability, meaning that we have some tumors will just have a ton of mutations, and there are drugs to handle that as well. And when we think about the way that we get cancer, we call it basically somatic versus inherited. Some people are born with the predisposition for cancer. Sometimes we talk about, and this is on this side here, germline mutations are basically, they'll be either in the egg or the sperm. They can be inherited. So a person would carry that mutation, and you can identify this in a person's blood. So it's in every cell of the body versus if you have what we call somatic mutation. Somatic mutations are found within the tumor. So my area that I study is somatic cancer, and that's my main focus. And I study somatic cancer in solid tumors. People can study cancer in hematologic malignancies. Right now, I'm focusing more on solid tumors. So anything besides cancer in the blood. And when we think about this somatic cancer, or some people call it sporadic cancer. So when we start off, when we're really young, we have cells that don't have a lot of mutations. And over time, we can start developing hits. And what we call them is sort of like insults on cells, where you develop them as the cells rapidly divide. Sometimes you'll just get a mutation. Then over time, you can have what we call a driver. So we call things passengers, where they're accumulating over time, but they're not really causing the cell to become cancerous. Once you have a mutation that is going to disrupt function of the cell to a certain level, that's where we call it a driver. And once you hit a driver, this allows cells to become what we say clonal. So they can then expand and make lots of those cells that have these mutations in them and cause a tumor. And so what I do is I study all the different variants that we would call drivers. We look at passengers as well, where we're able to use certain drugs that target lots of mutations versus the drivers where we can target those drivers with specific drugs that are designed. And so for what I do is I've actually put in my little Meaghan's focus, but basically we get samples at my company and at other companies that do tumor profiling. And tumor profiling is just basically you look at a tumor and you look at all the mutations in that tumor, whether it is on a large scale or a small scale. You could test a tumor just and look at 2 mutations, or you could test a tumor and look at your whole genome. And so what you would do is we get samples that are basically the tumor itself that they put into what we call paraffin. And that's what this looks like. It's a pathology sample. So after a person will have surgery, they put this into a special paraffin embedded. And then we take slices of the tumor. And Marilyn probably did or saw some of this work happen years ago.
Absolutely. Really very, very thin slices.
Very micron level, teeny slices. And we
Right.
realized, yeah, you get very little. The tumor is super precious. So you have to decide what you want to use to, you can't just go out and exhaust a tumor sample, because it can be, when we get it from a pathology lab, it is basically, we're going to use it, then we send it back for a different test to be done on that same tumor. So it's really precious. And that is what we would call, the tumor profiling component. But now we're also looking at, we're doing liquid biopsy, which is basically you can take the blood and we can detect tumors can shed basically cells that have these mutations in them, and that you can identify within the blood. And this is a newer advance that it's easier to look at. I mean, it's easier to look at blood because it's easier to collect. It's harder to get the tumor. However, your sensitivity of getting the actual mutations is better if you're doing the tumor. But then again, if you want to do multiple samples over time during progression of disease or in between treatments, liquid biopsy is what the goal is to be able to use. But what I do right now, our company is working on liquid biopsy, but we're working currently on the tissue collection, using the tissue. So what we do is we extract basically the DNA out of these tissues here. And what we do then is in my work, I have to say what genes are important for us to sequence that we know that are important in clinical care, whether they're diagnostic, prognostic, or there are drugs that can target this particular gene or dysfunction of this gene. So this is an example. This is not our panel, but this is just a kind of an accumulation of different images to show you sort of the workflow. But this would be an example of, we're looking at large panels of a couple, 500 genes that we look at. So you have to understand what are the roles of each of these genes, how do they function, how are they dysfunction, and then what happens in cancer. So what you do is you'd sequence the DNA for all of these different genes, and it's machine, and we call it next generation sequencing. It's just the term, because you can do it rapidly, it's less expensive, and over time, this is the way that this works. But you can also do, depending on, here we're talking about a panel of different genes. You can do the whole genome, you can do the exome, meaning only the coding regions of the gene. And it's more expensive the more genes you do, because it takes up more space on the panel. And then what happens is you sequence it. So we have all these different copies of these particular genes, and then we put it into these, what we call bioinformatic pipelines, where we can actually look at the variants in each of these genes. And so this is done computationally, and then it spits out. And what my role is I look at all of those variants that have been annotated just by saying, this is the gene, this is the location of the mutation. And then I go ahead and I say, how does that mutation impact the function of the protein? Is it relevant in causing oncogenicity? Is it oncogenic? And we have to understand, what does each protein do in cancer? Thinking about the different pathways of cancer. And so I analyze the variants, and then what we do is we want to generate a patient report. Patient report will have information about the variants, as well as the drugs that are applicable to treatment of these variants, and also response information as well as resistance information, because we also know that different variants will be associated with resistance to certain therapies. So you have to look at it in a profile, not just one specific gene at a time, because sometimes you'll have 3 or 4 mutations that together will say, oh, you can't have that drug. So, and that's kind of what I mean here, where I showed you this sort of the hallmarks of cancer. Each year, it expands. And so this is kind of a more specific one, but it's just meant to show that each of these different hallmarks, for example, proliferation or evading growth suppression, each one of those has genes that are associated with the function. So what we know is that you can have mutations in different genes that cause problems in different pathways. And we know that patients can start off with a particular mutation in one gene, and then cancer is tricky. So what it can do is you can kill off those cells that have these, that do this function inappropriately, and then your cancer will evade and then say, oh, I'm going to use a different pathway down here, and now I'm going to signal through this so that the cancer can still survive. And so what we do is we try to understand all the mutations the patient has, as well as different ways of how the cancer is trying to get around it through treatment. And that's why monitoring cancer is becoming can become in certain cases more of a chronic disease that we're treating versus blasting it with chemotherapy. Chemotherapy is usually the start, and then they begin with these targeted therapies, unless you have mutations in certain genes where you can target them right away. I'm going to give an example of how we think about lung cancer. And I'm thinking about one of the main types of lung cancers, non-small cell adenocarcinoma. And these are examples of drivers, meaning that these are the mutations that would happen that would cause the cancer to grow and metastasize versus just a bunch of passengers that accumulate. So when I see different variants like these, which you don't need to really see, but you see certain genes, for example, EGFR, or you see mutations in ALK, different ROS, we know that that's a driver. And what we have to do is say, okay, it's a driver gene, but what does that variant do? So that's my goal, is to say, what does that variant do, and how is it really driving the gene, or is it just a inconsequential variant in that gene? So there's a bunch of different, so every time you sequence a patient, we have to think about the cancer type when we are thinking about the how we're going to handle it. I figured I'd show you kind of a little bit of an example of how we see the treatment in terms of targeted therapy and how it's personalized. So for an example, we have a 63-year-old woman who has advanced lung cancer, which I'm still sticking with, the non-small cell lung cancer. And she started off with a baseline chest scan, a CT, and they found a mass. So this is her lung. This is right here. This is actually the left side of the lung. And you can see the circle here where you see the opaqueness. And this is where her tumor is. And what they did was they did testing. And when you have lung cancer, they often will do particular gene testing to see what mutations you have to determine the drug that you should get. And they found out that she had an EGFR particular L58R mutation, which is associated with response to a drug called erlotinib. So she was able to get this drug, and then 2 months after follow-up, another scan was done, and you can see that the tumor has regressed. So the EGFR tyrosine kinase inhibitors, which are a class of drugs, are really great in removing and getting rid of these tumors. However, the problem is that patients will then, the cancer will evade. So they'll develop resistance mutations, so that the drug that worked so well to bind the protein now no longer fits. So they developed a resistance mutation. And so down here in this blue, this is basically a crystallography image. And it's just meant to show that this little red here is the erlotinib drug in this protein. And you can see it's in here. And you can see here now that we have this mutation in here, which 15 months later, the patient develops this T790M, and now the drug doesn't fit. So the drug's not going to work anymore. So now they say, oh, and you look over here and you see there's mutations happening again on the other side. So usually it's about a year that the patient will do fine on, typically on a drug, which we call like a first line treatment. Then you give them another treatment. And so now, since this is growing, we notice that this drug won't work. Now we give a different drug. And so now 2 months after follow-up, you see that these tumors have regressed. And then 10 months after this, we see that another tumor is growing, because what happened is now a new mutation developed because of treatment with osimertinib. So this seems, it's like a constantly just keeping in check and switching drugs over time as the patient develops resistance mutations from the therapies that they're getting. And that is something that is very common in using these targeted therapies. And that's why that now for this patient, what would the option be? It would be they're testing different combination therapies. So particular drugs in combination work differently than they do by themselves, as well as they'll often add in different chemotherapies, and they're looking at drugs in clinical trials. So part of my role is also to be able to provide clinical trial information for each patient if there are no therapies that are approved in their cancer type or approved in a different cancer type. And this is just to show we know that there are for lung cancer, there's several drugs that are FDA approved and others that are in later stages of investigation. And here I'm just listing drugs. And we can see here that there are many mutations that these drugs target. And then there are many resistance mechanisms that are associated with these drugs, for which if we know you have a resistance mechanism, we would automatically not, we would write in the report, do not take this drug because this is associated with resistance. And that's what's most important also with precision medicine is really, or personalized medicine is really knowing what that patient, what would work and what wouldn't work. Because if not, you would be blind to that. And this is the kind of things that I look at when I'm sleuthing around. So a lot of it is kind of investigational skills because we read the literature, we have a huge knowledge base that provide, where we collect the cancer type, the disease, the drug information, as well as what we call biomarkers, which are these variants, biomarkers of response or resistance to therapy. And so we look at signaling pathways, we look at lots of dorky, lots of nerdy things, but it's, to me, it's fascinating. But we understand signaling pathways of genes that are, or proteins that are actually associated with development. A lot of them are associated with development early on, but they go haywire later, and that's where we see a lot of links to cancer. And many of these targeted therapies are important because if you were to use chemotherapy is a way to eliminate rapidly dividing cells. And so rapidly dividing cells are our hair, our skin, our inside of our mouth, all of those areas, which are also impacted when you do chemotherapy or you use that. So targeted therapies are known to have less side effects, although there are other, but they're risky, and they bring with them their own set of issues. So we really have to make sure that if you believe that a targeted therapy would work, given the research, given the literature, given our evidence-based review, that's when you would list a targeted therapy. And so for my final slide, these are not our reports because we're in the process of development and I didn't add them in here, but these are just other companies' reports showing kind of the end result of what we would do is we would have be able to provide the patient with a list of variants that they have. For example, this tumor has an ALK and a CCND1 mutation. We provide the list of drugs that would be associated with response and or with resistance. And the goal with my work is to be able to have a profiling report versus an individual row of each variant is by itself. It's really, you might have 3 variants that together equal almost a unit where you're going to describe a drug relationship. And so that's our goal. And yeah, and so that's what I do on a daily basis. Or
well, that's a high flying overlook, and it almost sounds like a game of whack-a-mole.
You're absolutely 100% right. That's what we do. It's whack-a-mole. It's just finding something else to keep managing the chronic cancer.
Do you ever meet a patient?
Not recently, more so now I'm more I meet oncologists and I speak with them quite frequently, just to be able to understand the needs. In the beginning, like in my previous work, I was with patients in my birth defect studies, but as I'm thinking now, no, not really. It's more oncology level that I'm dealing with or working with, getting input. And we're actually in the process of also thinking about how do we communicate the data to the patients as well as the oncologist. It's when we look at these reports here, they're confusing to a lot of the oncologists. So our goal is to create reports that are more understandable and not 50 pages long, which a lot of these reports are, and they're confusing. And so it's really being able to communicate with the physician as well as having a portal for the patients to log into and learn at their pace.
I could see that would be important because you want your patients to be compliant. You want them to understand. And it's not only the patient you're dealing with, but their extended family as well.
Exactly.
What kind of a team are you leading right now?
So my team encompasses, we have pathologists on my team, which we are molecular pathologists that do this case sign out. And we have 11 other people that are all PhD level trained molecular biology, structural biologists that are in the personalized medicine field. So we all work together and we are in a huge phase of development right now. That's why I'm not really showing my company's products because we are in the process of creating our own internal system. And I've been working on this for 2 years now on this project. And it's very painstaking, but the framework being built has to be so granular so that you can use the clinical data and the data coming out from the literature to be able to support every decision that's made with drug relationships.
Well, and you don't want to build it twice. You want to
Exactly.
that product.
We all came from other companies.
That everybody will accept at once. Absolutely.
Exactly. Yep.
I understand that quite well. When you talk about the scope of what it is that you're doing, when we're looking at this, how widely accepted is this? And where do your patients come from? Is this an international scope? Or are you primarily working within the health community of Boston?
We are working across the country. It's becoming more and more accepted where the drugs are getting approved on targeted therapies are getting approved more and more frequently. So, and a lot of them require, for FDA approval, they require to have molecular testing beforehand, before treatment. So it is becoming more accepted. And there are several companies that are doing hundreds of thousands of cases a year. As the field evolves, the expectation is that it's going to be more and more common. We see it largely at bigger centers. Smaller community hospitals are sort of the area where it's almost convincing that this information makes sense or it's important. But if you have Mass General, you have Sloan Kettering, all the larger cancer centers are all fully doing this work.
Now, this has been a long journey from when you graduated from high school at Western Reserve Academy. Was this even on your radar when you were a senior in high school, that this was going to be a path that perhaps you were going to end up at? I'm thinking that the field wasn't even around at that point.
No, I mean, I always wanted to be a doctor. So that was always since I was little, that was my goal, was to be a doctor. And so I was always interested in going to the hospital, visiting, seeing people in rooms. I mean, I was one of those kids. And so I always, I volunteered. I volunteered in high school, and then I volunteered at Mass General when I was in college, just working the ER and things like that. Yeah. But then I think, in college I did biology, which exactly I was doing it was so different. It was, PCR was brand new. Being able to do some of this work was amazing. And then the transition from where I am now, it's incredible. But I think just because I always wanted to be a doctor, I always knew, and then I didn't go into medicine, but I ended up kind of getting the clinical side of it by doing the birth defects research that I did and my work there. I still felt like I had that. And then doing precision medicine, to me, it's really fascinating. And I feel like I can help people on a day-to-day basis. And that's really rewarding. And yeah.
You made a reference to the flip side of the coin as well, and that is the family side, the genetic side. And we're hearing a lot about the 23andMe's and some of those commercial companies that are looking at those things.
Mm-hmm.
Do you have any comments about that prospect? As a consumer, is this something you're looking at?
I think it's being careful because 23andMe is fine. These companies are fine, but you have to also know that if you're using it for a medical device, which I know that you have to just understand that even if you're testing, BRCA mutation for breast cancer, they're not going to test every variant. So you might think, oh, I'm good, but really they're not giving you, it's not assessed by geneticists and people that are working and understand exactly what that mutation means. It can be a little misleading. So I would say if you're using it for a medical purpose, make sure that you think twice, or at least you understand what the limitations are.
In other words, you're saying it's a fairly rough screening mechanism?
Yes, it's rough and it's fun. I would say it's more fun to me. It's more fun to look and see, but I wouldn't use that to say, do I have risk for cancer, or do I have risk? Because there's also a lot of other things that are involved. It's not just a variant test as well.
Well, we've had a question come forward about the fact that we're hearing that some research centers are launching or testing a breast cancer vaccine. Do you know anything about this?
I don't know exactly about the vaccine, but they're doing a bunch of different vaccines. We see clinical trials frequently on particular colon cancer or particular mutations that they're looking at. I don't know which, I have to look into it, but yeah, I'm not surprised. It's interesting. It'll be interesting to see how it does.
Do you personally make lifestyle choices that might prevent cancer in your own family?
Can you repeat that? Sorry.
Do you personally make lifestyle choices?
Probably, I don't smoke. I don't drink. I don't know if it's just, I mean, I know the risk and I know that it's weird because there's so many ways cells can go wrong. And it's amazing how we actually do okay most of the time. And it's pretty fascinating, but I think, yeah, I try not to do any risky behaviors or things that are associated with smoking, all that kind of stuff. But also I feel like it's a predisposition. Like we probably have mutations in our body that we're born with that then you have that kind of second hit. So you might have a germline mutation and then you also might have an exposure or something that doesn't seem like it's a major exposure, but that's what puts you over the edge kind of. And we don't really know much about that. I mean, we're just learning. It feels like every week there's something that comes out and it's super quick moving.
Right. Now, the images that you were showing us in the example, the tumor,
Mhm.
those are all taken from CT exams. Is there a way to detect secondary mutations via the blood before that tumor would actually show up?
Yes. Yes. And that's the goal of the testing, the residual disease testing that we're working on as well, is where you would basically, in some studies show that you can identify that there is a change in the tumor, or there's basically, you're going to get a tumor several months to almost a year before that tumor comes back. So it's doing these tests and saying, okay, I'm going to test all the genes that we know that are associated with resistance, and I'm going to test that in the blood every couple of months, maybe 4 times a year or 8 times a year, and see, does anything change in my profile or my landscape of my tumor? So it's thinking about all these mutations in your tumor. What's my landscape? What's changing over time with my treatment? And then you might want to modify your treatment before you see a tumor because you notice that certain markers are changing.
I can understand. The sooner you get at it, the easier it would be for the patient.
And it's defining that. It's really understanding when is it a false positive, when is it real, because it is so subtle, and extracting the DNA from the blood, from the tumor. So there's a very fine line. So that's still being worked out by laboratories and really kind of fine-tuned.
How have you been impacted by COVID-19?
Well, I'm working in the basement right now, but I work from home. I work from a friend's house every day. It's one of those things where I think the company itself I usually work in Cambridge, but everyone works from home, the entire company except the laboratories it's based in California. They're all homework, but luckily we have a very good system of communication in Zoom and Slack that it keeps everything afloat. But I think that the community itself, in terms of testing, it went down with COVID in the beginning. Nobody was doing any testing. So my company is mostly germline testing. So they do all sorts of anything that's inherited, cancer as well as cardiovascular, a newborn, all these kinds of screening they do. So when everything shut down, the medical community, it went way down just because of no one was testing. So then what they had to do is become more resilient and flexible and figure out, okay, well, we can send out kits, we can have Zoom conferences with the patients. It's really becoming flexible and being able to get over that hump. And that's what they were able to do. But it was definitely a transition of learning how to function as a company.
Right. Well, you're a mom too. You've got 2 little boys.
Yes, 2 boys that are very active.
And sure, of course they're active, and they're home.
My mom says they're hard.
Yes. So how are they doing at school? What's their school experience?
Actually, they are going to school full time. We have them in 2 different Catholic schools nearby that are actually able to maintain the 6 feet distance and go into school. So it's been really great. And on occasion they'll be hybrid or doing remote when there are cases in the area or a case in a classroom or things like that. But we've been super lucky.
Yeah.
Because our community is remote. Or is hybrid.
Sure. Okay.
Yeah.
It's been absolutely fantastic. There's a company, Northwest Biotherapeutics, that are developing solid cancer treatments.
Mm-hmm.
They've been at it for about 14 years. Are you aware of this company? How many of these companies are out there?
That are developing like drugs, targeted therapy drugs, or?
Well, the question comes forward that they're developing solid cancer therapies. Are those like, Bill, please clarify, is that like a seed, implant seeds? Because I know for some therapies.
Some therapies, they do put radioactive seeds or particular chemo. But solid tumor, so it would be, solid tumor would be, there are drugs that are approved for all solid tumors. Maybe that's the question, where there are a few that are now approved for if you have a fusion where you have a protein that's created that's wrong, and basically you make a ton of this particular protein. So now that drug is approved for anybody who has that mutation, not just in one cancer type, it's approved for all solid tumors. So that kind of goes to show that sometimes it's not tumor specific, it's just the protein that went wrong, and it can be in any part of any tumor in your body, and that drug will work. So there are solid tumor drugs that are out there that they're being developed, but they're less, they're not as frequent, but they're great.
Now, everyone has got COVID on their mind, and the vaccines are rolling out now. Has there been an intersection between what you're doing and the vaccine creation?
My company's actually been able to, many genetic testing companies are able to quite easily create the testing in-house. They can develop it because it's not hard. Once you have platforms, you can do it. So we have that capability now, and they're actually doing testing internally as a pilot to be able to test employees who are coming in so people feel safe. And so they're doing that automatically.
Is this PCR testing?
PCR testing, yes. Okay. And so it's phenomenal how quick and flexible it is. Now we're able to also think about how, because it's been a situation where you think about viruses in general are important for development of cancer, of different diseases. And so that's always been on kind of the radar of how do we utilize, being able to test other viruses in the future, being able to handle it, being able to think about its role in disease as well. So that's always been on the table, and now it sort of promotes that in terms of people's radar of what the next step is for looking at disease.
As lay people, many of us have been absolutely amazed that the COVID vaccine was created this fast.
Yeah,
Did it surprise you as well?
Yes, I mean, that's phenomenal. I think it's enough money, enough manpower, because usually vaccines don't provide as much money because they have to be cheap to be able to disseminate and stuff like that. So this was a great way to be able to get there. And I know that people are talking now about what if they're the vaccine, what if people are resistant, what if there's new strains? And I think it's a matter of also understanding how the vaccine works, what it can and can't do. And I think time will tell. But yeah, I mean, it's the fear part too of, oh, this new strain won't work, or this, but I'm thrilled. I'm looking forward to getting the vaccine. So I'm waiting.
Well, it's been absolutely a joy.
Oh, thank you.
Joy to have you with us this morning. You give us hope.
Thank you.
You give us hope because cancer, I know that cancer is not one disease.
No.
That for many patients, for many people in the United States, I think it's one disease. It's very, very specific to that individual, and it always has been.
Yes.
Always has been. One type of breast cancer is not the same as another person.
Every person's different. Everyone handles treatment different. You can't assume that you're going to be the same as someone else who has the same stage, all of that stuff. It's very independent. And yeah.
And that's why this approach towards the treatment is very, very special. Mm-hmm. And I'm really pleased to see that it is here. It's finally
here. It's getting there. And I think we have a long way to go, but at least we're making that path forward. So.
And I'm so pleased to see that young people, bright young people from Hudson, Ohio, are involved with this project as well. Okay. Congratulations on your work. And I know you have a very bright future.
Thank you.
Your mother's proud. You need to.
She's the best mom in the world. So she did a good job.
Okay, well, that is pretty awesome. Well, we're going to start our close at this point, but thank you again for joining us. This has been absolutely terrific. Well, I'd like to remind people that we have two members who are kind of in sick bay. Liz Ramsey is still at Heritage of Hudson, but she's expected to go home soon. Ben Rexroad is already home after his successful surgery last Thursday, in fact. So Sandy's in charge again, and that's a wonderful thing. Next week, we're going to hear from Devi Gursahani with one master's degree in social work and another one in organizational development. She is now spending much of her time and her energy on a new project called Red Flag, an organization that is assisting our schools in identifying and finding solutions to the significant mental health issues facing our young people. And then we will close, as we always do, looking backwards in history. On this day, in 1841, China cedes Hong Kong to the British during the First Opium War. In 1971, the single record, What's Going On, was released by Marvin Gaye about police brutality. In 1980, President Jimmy Carter announces the US boycott of the Olympics in Moscow. And in 2008, Breaking Bad was created by Vince Gilligan, starring Bryan Cranston, and premiered on our TVs. So that's what's been happening in the past. Today is a momentous day, and let's all recognize it as such. So thank you again for joining us this morning, and thank you, Meaghan. This has been just a delight. I knew it would be.
Oh, well, thank you so much. It was a pleasure.
Yes, thank you. And Allison, unmute and let us know say hi to your daughter.
I'm saying hi, Meaghan, and also to her cousin Erin, who always wanted to be a nurse and is a nurse. She's on, and then my twin sister's on, and Meaghan's sister, Kara, is on. They're all listening and very proud.
Oh, boy.
Oh, and Kent is here too, Meg.
Oh, good.
Working in the background.
Hi.
That's a lot of pride for one Zoom call.
So we have a great family. lots of support.
Well, and I didn't even get around to asking you about the things that you were involved with in high school that really built this, other than the volunteering in the hospital.
One thing I did was Hattie Larlam. I remember that was it was like a service, and I went to Hattie Larlam, and I remember going and swimming with the kids who had disability. That to me sticks out. I don't know. It was just really neat listening to the other students talk about service and all that. We